Hello dear students, welcome to the biology online class. So today in this video we are going to study about the new chapter, that is chapter 2, sexual reproduction in flowering plants. Okay, in plants the flower, it is the structure which is meant for sexual reproduction. Okay, so flower is the structure which is meant for sexual reproduction because this flower contains the sex organs, and these sex organs are also called as reproductive organs. So all the flowering plants show sexual reproduction, and as I said before, flowers are the sites of sexual reproduction. Now, a typical flower, it consists of 4 parts. They are sepal, petal, stamen and carpel. So, all these 4 parts are present in a typical flower. Of these 4 parts, the first 2 parts means the sepal and the petal are referred as non-essential organs or accessory organs because they do not involve in the process of reproduction. Okay, whereas the stamen and the carpel are referred as reproductive organs or essential organ. Why? Because in flower these two structures are directly involved in the process of reproduction. So in a flower, the sepal and the petals are referred as non-essential organs, whereas stamen and carpel are referred as essential organs or reproductive organ. Okay, is that clear? Now coming to the reproductive organs. So in flower there are 2 types of reproductive organs, right? So one is stamen, which is also called as the male reproductive organ, and the second reproductive organ is carpel or pistil. Now coming to the first type of reproductive organ in the flower, that is stamen. So this stamen, it is also called as androecium. Okay, so in a flower, stamen is considered as the male reproductive organ, and each stamen consists of 3 parts. They are filament, connective and anther. Once again I'll repeat the point. Each stamen is nothing but it is the male reproductive part of the flower, and this male reproductive part of the flower is also called as androecium. Okay, now each androecium, it consists of 3 parts: filament, connective and anther. Now filament, it is the stalk like part. So in the picture, so this portion is called as the filament. So filament is nothing but it is a long stalk like part. Am I clear? Okay. Now anther means it is the terminal portion of the stamen, or the tip portion of the stamen is called as anther. Is that clear? So filament is the long stalk like portion, whereas the tip portion is called as the anther. Now in between the filament and the anther there is a connective. So which part is the connective here? So this part, this greenish color structure is called as the connective. Am I clear? So once again I'll repeat. Filament is the long stalk like part, whereas anther is the tip part where it contains the pollen grains, and connective is the part which connects the filament to the anther. Okay, so this is about the structure of a stamen. Am I clear? Okay. Now the anther may be monothecous or dithecous. Now anther is nothing but it is the tip part of the filament, tip part of the stamen, correcta? So this anther may be monothecous or dithecous. So monothecous means the anther may be unilobed. What do you mean by unilobed? Unilobed means single lobed anther. So now this is the picture of single lobed anther, and this is the picture of dithecous anther, means two lobed or bilobed anther. So the monothecous anther is also called as unilobed anther, whereas the dithecous anther is also called as bilobed anther. Is that clear? Okay. So when we take the section of this anther lobe, once again I'll repeat. So when we take the TS of this anther lobe, we can observe two chambers in each lobe. Okay, so in each lobe we can observe two chambers, so that a monothecous anther contains two lobes or two chambers, and these chambers are also called as microsporangia or locules or pollen sacs, so that a monothecous anther is said to be bilocular or bisporangiate. Once again I'll repeat. So when we take the section of the anther lobe, okay, so each lobe contains two chambers. Am I clear? Now the monothecous anther is unilobed. Am I correct? Yes. The dithecous anther is bilobed. Clear? So the unilobed anther, it contains single lobe, and when we take the section of this lobe, we can observe two chambers, right? So monothecous anther ಅಲ್ಲಿ ಒಂದೇ ಲೋಬ್ ಇರುತ್ತೆ. ಸೊ ಸೆಕ್ಷನ್ ತೆಗಿಬೇಕಾದ್ರೆ ಮೊನೋಥಿಕಸ್ ಆಂಥರ್ನ ಲೋಬ್ಸಲ್ಲಿ ಟೂ ಚೇಂಬರ್ಸ್ ಇರುತ್ತೆ, ಕರೆಕ್ಟ್ ಅಲ್ಲ? ಸೊ ಇನ್ ದ ಸೇಮ್ ವೇ ಡೈಥಿಕಸ್ ಆಂಥರ್ ಕಂಟೇನ್ಸ್ ಟೂ ಲೋಬ್ಸ್ ಇರುತ್ತೆ. ಸೊ ಸೆಕ್ಷನ್ ತೆಗಿಬೇಕಾದ್ರೆ ಈಚ್ ಲೋಬಲ್ಲಿ ಟೂ ಚೇಂಬರ್ಸ್ ಅಂತ ಹೇಳಿದರೆ ಟೂ ಲೋಬ್ಸಲ್ಲಿ 4 ಚೇಂಬರ್ಸ್ಗಳು ಕಾಣಲಿಕ್ಕೆ ಸಿಗುತ್ತೆ. ಆಮ್ ಐ ಕ್ಲಿಯರ್? ಸೊ ದ್ಯಾಟ್ ಮೊನೋಥಿಕಸ್ ಆಂಥರಲ್ಲಿ ಟೂ ಚೇಂಬರ್ಸ್, ವೆರಾಸ್ ಡೈಥಿಕಸ್ ಆಂಥರ್ಸಲ್ಲಿ 4 ಚೇಂಬರ್ಸ್ಗಳು ಕಾಣಲಿಕ್ಕೆ ಸಿಗುತ್ತೆ. ಇದು ಕ್ಲಿಯರ್ ಆಗಿದಾ? ಓಕೆ. ಈಗ ಈ ಚೇಂಬರ್ಸನ್ನು ಲೊಕ್ಯೂಲ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ದ ಚೇಂಬರ್ಸ್ ಆರ್ ರೆಫರ್ಡ್ ಆ್ಯಸ್ locules or microsporangia or pollen sacs. Okay. Now, because of the presence of two chambers, the monothecous anther is also referred as bilocular or bisporangiate anther, whereas a dithecous anther contains 4 locules or 4 chambers, so it is said to be tetralocular or tetrasporangiate. Is that clear? Okay, we'll move on to the next slide. Now coming to the structure of microsporangium. What do you mean by microsporangium? Microsporangium is nothing but the chamber of the anther lobe, which is also called as the pollen sac or locule. Now coming to the structure, a typical microsporangium is nearly circular in outline, and it is surrounded by 4 wall layers. They are epidermis, endothelium, middle layers and tapetum. So all these are the wall layers of a microsporangium. Am I clear? Okay. Coming to the structure. Now in this picture, so this is the structure of TS of microsporangium, TS of anther, where we can observe 4 chambers. Okay, these 4 chambers are called as microsporangia, right? Now each microsporangia, it contains 4 wall layers. The outermost wall layer is called as epidermis. So this is the epidermis, and epidermis is nothing but the outermost layer of the microsporangium. Am I clear? And it is a single layer structure. Now next to the epidermis, the microsporangium contains the second layer, that is endothecium. So epidermis is the outermost layer. Next to the epidermis, endothecium is present. Now next to the endothecium, middle layer is present. So middle layer is the third layer of the microsporangium. Am I clear? The outermost layer is epidermis. Next to the epidermis, it contains endothecium, and both the epidermis and the endothecium, they are single layered structure. And next to the endothecium, a double layered structure is present, or 2 to 3 layered structure is present, which is called as middle layer. And the fourth innermost layer of the microsporangium is called as tapetum. So tapetum is nothing but the fourth innermost layer of the microsporangium. Is that clear? Okay. Now the first 3 wall layers, the outer 3 wall layers, give protection and help in dehiscence of anther to release the pollen grains. So what is the function of epidermis, endothecium and middle layers? Then these layers give protection and helps in the splitting of anther to release the pollen grains. Whereas the function of the innermost wall layer, that is tapetum, is it gives nourishment to the developing pollen grains. Is that clear? So this is about the structure of TS of microsporangium. Once again I'll repeat the structure. So each microsporangium, it contains 4 wall layers: epidermis, endothecium, middle layer and tapetum. The first 3 wall layers give protection and help in dehiscence, or help in splitting of the anther to release the pollen grains. Whereas the function of the tapetum is to give nourishment to the developing pollen grains. Is that clear? Now, next to the tapetum, the microsporangium, it contains a group of tissue that is called as sporogenous tissue. That means at the center, each microsporangia contains sporogenous tissue. Okay, and this sporogenous tissue is composed of mass of cells, and each cell is here called as sporogenous cell. And this sporogenous cell is diploid in nature. Is that clear? Okay, once again I'll repeat. In a young anther, each microsporangium has sporogenous tissue at the center. All these are the sporogenous tissue. Is that clear? Now, this sporogenous tissue is composed of compactly arranged diploid cells, and each of this cell is called as sporogenous cell. Okay, and remember, always this sporogenous cell is diploid in nature. Is that clear? So these are about the structure of microsporangium. Clear? Okay, move on to the next slide. So this is the picture of TS of microsporangium, TS of anther. And this you can observe the layers, epidermis, endothelium, middle layers, as well as tapetum. And next to the tapetum, it contains the sporogenous cells. Okay. Now what happens? As the anther develops, each sporogenous cell, which is also called as microspore mother cell or pollen mother cell, undergoes meiosis to form microspore tetrads. Am I clear? Once again I'll repeat. See, next to the tapetum, sporogenous tissue is present, correct? So in this sporogenous tissue, it is composed of mass of cells, and each of this cell is called as the sporogenous cell, okay, or spore mother cell or microspore mother cell. Now what happens as the anther develops? This sporogenous cell, it is diploid in nature, right? This undergoes meiosis to form 4 microspores. Understood? So microspore mother cell ಏನಿದೆ which is diploid, ಅದು ಮಿಯೋಸಿಸ್ ಆಗುತ್ತೆ. ಮಿಯೋಸಿಸ್ ಆಗಬೇಕಾದ್ರೆ 4 ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ಗಳು ಫಾರ್ಮ್ ಆಗುತ್ತೆ. And all these microspores are haploid in nature. Correct? And these microspores are arranged in a group so that it is called as tetrad. They are arranged in a cluster of 4 cells. 4 cells ಒಟ್ಟಿ ಕ್ಲಸ್ಟರ್ ಅಲ್ಲಿರೋದ್ರಿಂದ ಮೈಕ್ರೋಸ್ಪೋರ್ ಟೆಟ್ರಾಡ್ಸ್ ಅಂತ ಹೇಳಿ ಕರಿತೀವಿ. ಓಕೆ, and the process of formation of microspores from the pollen mother cell through meiosis is called as microsporogenesis. ಈ ಪ್ರಾಸೆಸ್ ಏನಿದೆ, ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಅನ್ನು ಫಾರ್ಮ್ ಮಾಡುವಂಥ ಪ್ರಾಸೆಸ್ ಏನಿದೆ, ದ್ಯಾಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನಿಸಿಸ್. ಓಕೆ, ಸೊ ದ ಪ್ರಾಸೆಸ್ ಆಫ್ ಫಾರ್ಮೇಷನ್ಸ್ ಆಫ್ ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಫ್ರಮ್ pollen mother cell through meiosis. Okay, now what happens after microsporogenesis? See, ಇಲ್ಲಿ ಕಾಣ್ಬೋದು ನೀವು. Now this is the pollen mother cell or the sporogenous cell which is diploid. ಮಿಯೋಸಿಸ್ ಆಗುತ್ತೆ. ಮಿಯೋಸಿಸ್ ಆಗಿ ಈ ರೀತಿ 4 ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ಗಳು ಕ್ಲಸ್ಟರಲ್ಲಿ ಫಾರ್ಮ್ ಆಗಿ ಸ್ಪೋರ್ ಟೆಟ್ರಾಡ್ಸ್ ಆಗುತ್ತೆ. ಲೇಟ್ರ್ ಆನ್ ಆ 4 ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ಗಳು ಸೆಪ್ರೇಟ್ ಆಗುತ್ತೆ. ಅಂದರೆ ಮೈಕ್ರೋಸ್ಪೋರ್ಸ್ ಆರ್ ನಥಿಂಗ್ ಬಟ್ ದ ಪೋಲನ್ ಗ್ರೇನ್ಸ್. ಕರೆಕ್ಟ್ ಅಲ್ಲ? ಸೊ ಇದು ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್ ಅಂತ ಕರಿತೀವಿ. ನಾವು ಈಚ್ ಆಫ್ ದಿಸ್ ಮೈಕ್ರೋಸ್ಪೋರ್ ಈಸ್ ರೆಫರ್ಡ್ ಆಸ್ ಪೋಲನ್ ಗ್ರೇನ್. ಆ ಮೈಕ್ರೋಸ್ಪೋರೇ ನೆಕ್ಸ್ಟ್ ಏನಾಗಿ ಕನ್ವರ್ಟ್ ಆಗುತ್ತೆ, ಡೆವಲಪ್ ಆಗುತ್ತೆ, ಪೋಲನ್ ಗ್ರೇನ್ ಆಗಿ ಡೆವಲಪ್ ಆಗುತ್ತೆ. ಇದಿಷ್ಟು ಕ್ಲಿಯರ್ ಆಗಿದಾ? ಓಕೆ, ಕಮಿಂಗ್ ಟು ದ ನೆಕ್ಸ್ಟ್ ಪಾಯಿಂಟ್, that is the structure of a pollen grain. So here pollen grain is also called as male gametophyte. Why? Because this pollen grain carries the male gametes. Okay, so male gametes ಇರೋದ್ರಿಂದ pollen grain ಅಲ್ಲಿ the pollen grain is called as male gametophyte ಅಂತ ಕರಿತೀವಿ. ಸ್ಟ್ರಕ್ಚರ್ ಅಬ್ಸರ್ವ್ ಮಾಡೋದಾದರೆ, a pollen grain is nearly spherical in outline and it measures about 25 to 50 millimeter in diameter, and it has a 2-layered wall, exine and intine. Okay, nearly spherical in outline, round shape allirutte, and it measures about 25 to 50 millimeter in diameter, and each pollen grain has 2 wall layers. The outer wall layer is called as the exine and the inner wall layer is called as the intine. Clear? Now, the exine is the hard outer layer made up of sporopollenin. It can withstand high temperature and strong acids and alkali. So, exine has apertures called germ pores where sporopollenin is absent. Okay, now exine ಅಂತ ಹೇಳಿದರೆ, in the pollen grain the outermost layer is called as the exine. Okay, and this layer is very thick due to the presence of sporopollenin. So sporopollenin is the wall material found in the exine of the pollen grain. Is that clear? Now, at some places This exine region of the pollen grain is remain thin walled ಡ್ಯೂ ಟು ದ ಆಬ್ಸೆನ್ಸ್ ಆಫ್ ಸ್ಪೋರೋಪೊಲನಿನ್. ಅಂದರೆ ಕೆಲವು ಕಡೆ ಏನಾಗುತ್ತೆ, ಒಂದೆರಡು ಮೂರು ಕಡೆ ಎಕ್ಸೈನ್ ರೀಜನಲ್ಲಿ ಸ್ಪೋರೋಪೊಲನಿನ್ ಡಿಪಾಸಿಟ್ ಆಬ್ಸೆಂಟ್ ಆಗಿರುತ್ತೆ. ಸೊ ಆ ರೀಜನ್ ಏನಾಗಿರುತ್ತೆ, ನ್ಯಾರೋ ಆಗಿರುತ್ತೆ ಅಥವಾ ಥಿನ್ ಆಗಿರುತ್ತೆ. ದೀಸ್ ಥಿನ್ ಏರಿಯಾಸ್ ಆರ್ ರೆಫರ್ಡ್ ಆ್ಯಸ್ ಜರ್ಮ್ ಪೋರ್ಸ್. ಏನು ಫಂಕ್ಷನ್ ಜರ್ಮ್ ಪೋರ್ ಇದ್ದು? It helps during the germination process to form the pollen tube. Okay. So, idishtu clear agida? So, this is the outer layer which is called as exine, which is made up of sporopollenin. And at some places the sporopollenin is absent and that area remains thin walled, which is called as germ pore. And during germination, a germ tube is formed through this germ pore. Clear? Now, coming to the inner wall layer. So, the inner wall layer is called as intine, and this intine is composed of cellulose and pectin, and hence it is thin. So, exine is thick because it contains sporopollenin, whereas intine is thin because it contains cellulose and pectin. Am I clear? Now, next to the wall layers, cytoplasm is present, which is surrounded by the plasma membrane, and a matured pollen grain contains two cells. They are vegetative cell and generative cell. A vegetative cell is bigger and it has a abundant food reserve and a large irregularly shaped nucleus, whereas the generative cell is small and it floats in the cytoplasm of the vegetative cell. It is spindle in shape with dense cytoplasm and a nucleus. That means, so this is the structure of a matured pollen grain, and this matured pollen grain, it contains two cells. The bigger cell is called as the vegetative cell, and the vegetative cell, it has its own nucleus, that is vegetative nucleus. Okay, and the smaller cell is called as the generative cell with its generative nucleus. Okay, now the vegetative cell, it stores plenty of reserve food materials. Am I clear? And it contains a large irregularly shaped nucleus, whereas the generative cell, it is spindle in shape and it is small, and it also possesses a single nucleus. So these are about the structure of a pollen grain. Am I clear? Okay. Okay, now over 60% angiosperms shed their pollen grains at two-celled stage. In others, generative cell divides mitotically to give two male gametes. Thus, pollen grains are shed at three-celled stage. Okay. The shed pollen grains have to land on the stigma before they lose viability. And the viability period of the pollen grains is variable, which depends on the temperature and humidity. Viability of pollen grains of some cereals is 30 minutes, and in some members have viability for months. These pollen grains are rich in nutrients, and pollen tablets are used as food supplements. Pollen tablets and the syrups increase the performance of the athletes and race horses. And pollen grains of some plants, for example, carrot grass, that is Parthenium, are allergic for some people, and it leads to chronic respiratory disorders like asthma, bronchitis, etc. So these are about the male reproductive organ of the flower, that is about the structure of stamen, the process of microsporogenesis, and the development of male gametophyte. Thank you.